Good morning and welcome to Global Healthcast, brought to you by Global Health Press. In this podcast series, once a week, we bring to you news and views about public health, infectious diseases, vaccines, and vaccination. I'm Joe Schmitt, and with me in Switzerland is Dr. Melvin Sanicas. Good morning, Melvin.
Good morning, Professor Schmidt, and good morning, good afternoon, or good evening to those who are watching us or listening to us today.
Our topics today is post-COVID syndrome. Why does it happen in children? We report about a new malaria vaccine that is showing protection. We look at a WHO approved second mpox vaccine, and I will speak of reduced doses or reduced schedules of pneumococcal conjugate vaccines. Let's start with rare condition after COVID, which is this post-COVID syndrome. And Melvin, you tell us all about it.
Yes, so during the COVID-19 pandemic, while severe SARS-CoV-2 infections in children were relatively rare compared to adults, approximately 1 in 10,000 children develop multi-system inflammatory syndrome, or the MIS-C. They present with symptoms like rash, swelling, nausea, and systemic inflammation weeks after infection. And this study, an international study led by the Imperial College in London, identified rare variants in the BTNL8 gene, which helps regulate gut lining immunity as a key factor, increasing the risk of developing MIS-C by fourfold in children. So, using advanced genome sequencing techniques on 154 MIS-C cases from Europe and the US, researchers from the Imperial College London uncovered that these genetic variants likely made the gut more sensitive to SARS-CoV-2 and eventually triggering systemic inflammation. And so this study is important because understanding the genetic basis of MIS-C provides critical insights into its development, risk factors, and potential treatment strategies, paving the way, of course, for better care and outcomes for these affected children.
So the question then is, what is responsible for MIS-C, multisystem inflammatory syndrome in children? So number one, it is COVID, right? And number two,
Mhm.
it is your genetic makeup. So there are two conditions that need to come together to cause the disease in the pathogenesis of the disease.
Mhm. Yep, and probably this is only one of the pathways. There could be others.
Okay. So there's more about that.
Mhm.
Interesting. Yeah. Look, there's always some learning, and I think in the future we will report more frequently about diseases that need a specific genetic background to result in a certain phenotype. We will learn this more in the future.
Yeah.
Yeah. Melvin, you have another interesting study on a new malaria vaccine. What is this story?
Yes, this is a new promising malaria vaccine approach. A small clinical trial conducted in the Netherlands tested a genetically modified Plasmodium falciparum parasite GA2, demonstrating favorable safety immune response and an 89% protective efficacy against malaria infection. So this is an innovative late liver stage design. The GA2 parasite was engineered to develop further in liver cells, exposing the immune system to a broader range of antigens, leading to enhanced immune response compared to traditional vaccines. And the study involved 25 participants, so just a small trial, right? Divided It divided into GA2, GA1, and also placebo groups, with the GA2 showing superior efficacy, no breakthrough infections, and comparable mild side effects to the control group. So really good news in terms of safety and efficacy. And the mechanism of enhanced immunity being proposed by the researchers is that GA2 induced significantly higher levels of polyfunctional CD4 T cells and produced pro-inflammatory cytokines, suggesting that cellular immune responses, rather than antibodies alone, really drove the observed high-level protection. And so now the question is, what is the significance of this study, right? So with malaria causing still over 200 million cases annually, this study highlights the potential for this GA2 as a novel, more effective vaccination strategy. And this definitely warrants further evaluation in larger and more diverse populations.
Melvin, I like the result and I like the learning coming from practice. I don't know how to expose children to 50 bites of a mosquito to get a protective efficacy of 50% of against malaria, 89% against malaria. Um, I guess this is all experimental at this point, and they need to sort it out a little bit further in the future, right?
Yes, it's a small study, and I'm sure these challenges that you are seeing at the moment, I'm sure they will be looking into it as they progress into the development of this vaccine.
Correct me if I'm wrong, the current vaccine that is out there has a protective efficacy, if I remember correctly, of 30%. I may be wrong
Yes,
here,
it's
but this 89
30s,
is then
30s. Yeah,
30.
30s. Yeah.
This 89 is 3 times higher protective efficacy, so this is really a breakthrough from
Mhm.
in principle, right? So the
Yes,
question
correct.
is how to get this GA2 malaria organisms in the right way into the body for protection. I mean, 50 bites with a mosquito, that is difficult to do, right?
Mhm.
But particularly, you cannot do it in a mass vaccination campaign, right? Not
Mhm.
possible. So this is something to be sorted out, but the principle is really, really promising if you get it into practicability, right?
Mhm. Correct.
Yeah. Very good. Very nice stories you have today. And then there's another one that is very interesting. Report about mpox vaccine.
Yes. So the WHO recently licensed, well, not really licensed, but gave the EUL, right, that emergency use listing for this LC16M8 vaccine. It's a live attenuated vaccine. They granted the EUL, making it the second vaccine supported by the organization WHO to combat the ongoing mpox public health emergency of international concern. So the Japanese government has pledged 3.05 million doses of this vaccine, along with specialized needles to the Democratic Republic of the Congo, which is the hardest hit country in this current mpox outbreak. And they have recorded over 39,000 suspected cases and over 1,000 deaths already. So this LC16M8 vaccine is recommended for individuals over 1 year old as a single dose using a bifurcated needle. However, it is contraindicated for pregnant individuals and immunocompromised persons, as this is a highly attenuated vaccine, right? It's also contraindicated for those with advanced HIV or those in immunosuppressive therapy. So the WHO emphasized vaccines as part of a broader outbreak response for this current mpox outbreak. It also includes improved diagnostics, infection prevention, treatment, and community engagement, of course. And they continue working with manufacturers and partners to ensure vaccine availability. And this LC16M8 vaccine has, of course, shown to be safe and effective in previous outbreaks in Japan. And just a reminder for everyone that the other pre-qualified vaccine, the MVA-BN from Bavarian Nordic, is for use in individuals aged 12 and older.
So it is always good to have more than one or two products available. The more, the better.
Exactly.
Whenever there is a concern about one vaccine or not enough product available, the more you have, the better it is, right? So this is good news in principle, and it is also nice that it is available right now. And yeah, tell us a little bit more. The platform is a live attenuated vaccine, right? It
Yes,
is it is
correct.
a live vaccine, a whole virus live vaccine, right?
Correct. It's a highly attenuated vaccine. Yeah.
Yeah. It would be nice to get a little bit more information on the characteristics of the vaccine, like what is the cell line that produces it in, details on the spread of the vaccine. If you apply it, it may spread to others that are unvaccinated one way or another. But it's very interesting in principle, and particularly that there is already an emergency use license by WHO. Really good, really good. Yeah. Melvin, I'm covering only two stories today, both published in the New England Journal of Medicine from yesterday, actually from Thursday this week. And this is one study where they look at protection, at seroprotection, induced by reduced doses of PCV13 or PCV10 compared to the full dose. Now, I start with a concern and with some disclaimers. The disclaimer is they use the term, or the term behind this is seroprotection. And that is a licensing term, and it means if a subject after vaccination reaches this titer in antibody response, then you are protected. And this assumption is certainly wrong. You may have a higher titer or a lower titer and you're not protected or still protected, or you may have no titer and you're protected, or you have a very high titer and you're not protected. Anything is possible. So the term is a little bit misleading to begin with, but it's a licensing term. And if you reach the 0.35 milligram per ml, if you're above it, that means then you are then you get a license basically. Then you can do it. Then you're safe. And a second disclaimer here is the 0.35 is wrong because each of the pneumococcal serotypes has a different titer that would be considered as a correlate of protection. So the titer for serotype 3 is skyrocketing high, and the 114 may be medium low. And this is not reflected by having 0.35 for all. Next point of disclaimer, PCV10 has a license in Europe and only in Europe based on a lower titer, which is 0.2 microgram per ml and not the 0.3 microgram per ml. And that is licensed only in Europe. Other licensing agencies did not accept this lower threshold for licensure. In any event, let's look a little bit at the data. What you see in the top line is for PCV13, 40% dose versus full dose. You see less than 10% did not reach the 0.35 ml. And if you use 20%, the situation is worse. So here, particularly with serotype 3, 23F and 6B, the point estimates of the endpoint are not met. And for PCV10, again, you see the titers are here. I'm sorry, for PCV10, the same is shown down here. And again, very similar picture. You get lower titers, but after all, it seems quite acceptable. After the booster, again, there seems to be an acceptable threshold for the licensing criteria. You see, this is 0.5 that was predefined, and then if you go to PCV10, it's a little bit more shifted to the left side. So again, it is what it is. It's a serological endpoint. And what does it mean if you reach or do not reach the titer if the 0.3 can be debated, right? You can say 0.2, you can say 0.235. You would have to go by each single serotype and we have to look at the protective or the correlate of protection for each single serotype, which is not done in this study. And it gets too complicated because these titers or protection were defined by Public Health England for PCV13 only. So after all, this shows you can use a reduced dose. You get a somewhat lower immunogenicity depending on each serotype. Now, the big challenge for this study is, does this relate to protection? So what is the biological implication? And now you need to know and understand one other principle of vaccinology, and that is no carriage, no disease. So a very good endpoint for pneumococcal conjugate vaccines is carriage. If your vaccine reduces carriage or acquisition, that's all you need. That is an efficacy study, because if you don't carry the organism, you cannot get down with the disease. That is the very short and simple point. So what the authors did in addition to this, let's look at the immune response, they looked at carriage rates. And now you can go here, PCV13 full dose, 2+1 schedule, 40% dose or 20% dose, the same for PCV10, 40%, 20, and full dose. And then there you can go to all the details here. You should read the paper. What I worry about is emerging serotypes, particularly those that need high geometric mean concentrations to prevent disease, and that are serotype 3, 6A, and 19A. And what you want to know is there emerging of a serotype that was not there before? Let's look at 3, 6A, and 19A, and 6A, 19A. The two serotypes down here are not part of PCV10, but the company claims, and it is in the European label, that the vaccine prevents infection with these serotypes. If you look at the carriage rates, depending on the dose for PCV13, carriage rates are 12, 10, and 9%, so between 10 and 12%. And you see already for PCV10, these carriage rates are higher. And if you go to 6A and 19A, which supposedly are cross-protecting, it is 10, 6, and 5, so between 5 and 10%. And here you have 12, 16, and 12, so this is much higher. So the concern is in the long run that there is emerging of some strains that may not be covered by the vaccine, and that cause you more trouble than if you would not use the vaccine. But still, I think overall it is a nice way to approach how to make more out of your vaccine money. And this is the point of the whole study. This is a study that was done in Kenya, in Kilifi, in a vaccine study site, and they pay like $3 for PCV13. And by using a reduced dose PCV13, they could vaccinate many more children of their 1.5 million population. So this is really a very important cost issue. Do I give a little bit less protection to all children in my country at a lower protection rate, but vaccinate all? Or do I vaccinate all with the best protection rate? And I think, coming from a Western country, this is a very nice study to address the problem for cost saving. And it is an urgent issue to be addressed. And we have to follow the thinking in these countries to get as many children protected as possible. Melvin, any comments on this one from your side?
Yes, Professor, I like the last statements that you made, because I think for us who are living in developed countries, this will not really be appreciated. But for countries that are in the developing world, this is really an important thing for them to be able to vaccinate more children, even though the protection, as you say, could be a bit less than giving what is the recommended dosage.
Yeah. In the New England Journal of Medicine, there was another article, and these authors used a different approach. And again, the principle is pneumococcal carriage and vaccine serotype carriage in children done in Vietnam. So the endpoint is not the immune response, but carriage. And I just can reiterate, no carriage, no disease. So it is a very relevant endpoint. Now in Nha Trang in Vietnam, it's an area where PCV had not been used before, and they did a PCV10 catch-up campaign. And the vaccine was offered to children under 3 years of age according to 4 schedules. The lowest one would be 1 dose only at age 12 months, 0, P, and 1. P is primary. Or 2 and 12 months, like what they do in the UK, you give 1 primary dose to give some protection early in life, and then you give a booster dose late in the first year of life, 12 months, or at the end of infancy, and then this is basically a booster dose and you get long-term protection. Or you reduce a regularly licensed schedule 2 for 12 months, 2 primary and 1 booster dose, or you use 3 primary doses and no booster, which I think is not a good idea. We know from the UK and the HIP experience it didn't work. So anyway, this is what they did, and now they did annual carriage service in infants 4 to 11 months of age and in toddlers 14 to 24 months of age. And the study was started in 2016. So they have now the follow-up for 4 years. And let's look at the top line here. For any serotype, you can see that carriage goes down, and that includes the unvaccinated, right? That is interesting. The unvaccinated do, however, on the PCV10 serotypes have a higher carriage rate that you can see here. And the vaccinated children, all groups have a lower rate of carriage. And serotype 6A, there is not much of a difference. And that is one of the problem serotypes apparently for PCV10. Let's go for toddlers. Here again, you see there is a higher rate initially of carriage in toddlers and the unvaccinated. And here you see a difference again in unvaccinated and vaccinated, and it looks good for all different groups. And serotype 6A is a little bit less impressive. Now, if you look at percentage of carriers who carried pneumococcal vaccine serotypes, PCV10 serotypes, again, you see a difference here with regard to all serotypes, and less impressive for serotype 6A. I think this is, again, a very nice approach. You don't need much of a laboratory, and you don't need to interpret any serology data. Again, no carriage, no disease, and I would bet it looks to me as if any of the schedules that they use is working well. I would prefer the 2-4-12 if you have the money, or the 2-12, because you protect children early on. Carriage is one thing. Disease is more rare and more difficult to capture. The burden of disease in the first year of life, in the first year of life, the pneumococcal burden of disease is high. And roughly 100 per 100,000 is the rate of invasive pneumococcal disease. So this is 1 out of 1,000, and that is what you want to prevent, right? That is severe disease that may damage for the rest of life. In addition, all children in the first year of life probably have an episode of otitis media. So you want to prevent early on. So if at all possible, the 2-12 would be my favorite. But maybe even the 12-month would work. It depends a little bit on the burden of disease in a given country, what to do best. Melvin, any comments on the second study?
I think this is really innovative what the researchers in Vietnam did, because it gives them these different potential options so that they could really play around with a schedule that would benefit their locality. But I think it's also similar to the first study, right? It's really a developing country trying to make use of the tool that's out there, but also looking at the fact that they could not really be following the recommendation of all these different doses. And I think it's an innovative way of looking at things.
Yeah. Look, we see this again and again in a clinical trial. You develop a vaccine, it is 1 billion you need to spend to get a new vaccine licensed. You want to be on the safe side, and probably we're overdosing and overscheduling vaccines a little bit to make sure the 1 billion is not lost because the schedule was not appropriate or the dose was not appropriate. After it is licensed, you can try to find out to reduce doses and schedules, right? But only after licensure, because before you don't know. Once you apply with it, and the dose was too. Once you start the developing process and your dose is too low, the vaccine is lost. All your investment is gone. This is the mechanics behind it.
Yep.
Melvin, I think this is the end of our topic today. Again, reduced vaccination with a single primary and booster dose was non-inferior to alternative schedules in protecting against carriage in infants and toddlers. And the limitations, in my view, is 19A. What is long-term surveillance? Not discussed here today. What we did discuss today is MISC and the pathogenesis and how the genetic background results in a disease. Melvin spoke about a new malaria vaccines, an experimental one that works nicely in an experimental setting. We talked about an WHO EOL approved mpox vaccine, and I spoke in my last presentations on immunogenicity of reduced doses or carriage with reduced schedules. Melvin, in the end, you have a funny of the week.
Yes, this is a not really funny, right? Because it makes sense. So the smarter we get, the more we just focus on things that we know. Because the more you know, the more you know that you don't know many things, right? So I think it, in a way, it humbles you. So you stop talking about everything and pretending to be an expert in everything. But it also, it really helps you see that there is so much to learn out there.
Yeah. Yeah. So I guess you're outspoken about opinions if you know not much, right? The less you know,
Yes,
the more you have an opinion, which has no substance, right? And if you
exactly.
know too much, in the end, you may be blocked from doing something because you say, I need more information.
Hmm.
It is an interesting concept here. Yeah.
Yep.
Melvin, thank you very much. And you have the last word.
Yes, thank you for listening or watching us, everyone. Stay safe and see you again next week.